Welcome back, everyone. We have a very exciting afternoon and final session today for you that is going to cover new. I'm hearing my background noise, but that is going to cover new and emerging technologies. Our first talk of the afternoon is by Dr. Marcy Glixman. She's the head of biology at Enclair Therapies, and she will be talking to us today about why you want to use stem cells for drug discovery. Thank you, Marcy, for being here, and looking forward for your talk.
Great. Thank you, Serene, and thank you for the invitation to talk about one of my favorite topics. And so I titled this because I'm hoping to convince you that why you want to use stem cells for drug discovery. So I'm going to highlight different areas in which it's applicable, and really it's been gaining a lot of use, particularly lately, more and more use and more acceptance in the area of drug discovery. So just right up front to tell you sort of what the take-home messages are. And after this, you can leave. No, but so, stem cells provide human cells, which is nice because a lot of cell lines are not human. So they're human cells that haven't been immortalized, and they are not derived from a tumor. So they're more likely to be more closely aligned with actually human, biochemistry and human molecular biology. So that's really probably one of the strongest things. And also, they can be used in multiple areas of drug discovery. And as I said, I'll highlight some of their uses. They can also be manipulated, to look at mechanistic studies. So if you want to create, known mutations that are found, or combinations of mutations, or you can adapt them for that. They can be studied with electrophysiology, so they become fairly mature enough for those type of studies. There's a lot of case studies now. In fact, also assay guidance manuals starting to put out some chapters associated with use of stem cells. And at the end, I actually have a list of resources that are great, for learning more. Because actually, it's a huge field. I'm not going to cover it all. I'm just going to touch on some points, and then there are some great resources to do deeper dives into any of these areas. Stem cells themselves can also be used as therapeutics, and I think this is another exciting area in terms of cell therapies. And really, I think it's going to become more and more apparent as more people use them that they can be used to accelerate the process of drug discovery, as well as our understanding of disease. So this is, I'm sure a number of people have shown some versions of this slide, but just to highlight that they can be applied to multiple stages. So from early on, in terms of target identification and validation, they're really useful as tools to look at the association of particular phenotypes, association of certain genetics with diseases, the ability to use siRNA on them to help validate targets in particular. They can be used for high throughput assays, and there's a number of examples of that in the literature. And you can even just do a sort of pilot screen with them in terms of, how likely is it for the endpoints you've designed, how likely is it for you to find compounds that might have the activity you're looking for. So for target ID and validation, and then for hit identification, as I said, you can use them for primary screens, also for secondary screens and ways of confirming if you have a cell line that's more going to be easier or higher throughput. Also for initial structure activity relationship, and all the things associated with hit identification. Also hit to lead optimization, being able to, especially if you're working on a certain disease and have a cell model as a a way of guiding and optimizing your compounds is another area. And then, of course, identifying a candidate. And then, the other aspect is around early development. So people have been using stem cells to generate models for different toxicology purposes. And I'll touch on that as well. And then finally, in the clinical world, ways of using cell responses, stem cell responses from disease patients to try to identify potential responders and non-responders for your drug of interest. So helping potentially segment the patient population better so that you can have better targeted drugs as another area. So the two major properties of all stem cells that provide the advantages of working with them is one is that their ability to go through multiple rounds of cell division and retain that undifferentiated state. So you have a whole pot of cells, many vials that you can freeze away that are where these cells have the great ability to just proliferate without with any modification, just this undifferentiated state. And then the second property is really their ability to differentiate into specific specialized cell types. And that, scientists have made probably at least 200 or more different cell types from stem cells. So this is really useful in terms of, properties and again, in terms of diseases, knowing and understanding particular aspects of disease based on differentiated cell types, and I'll be giving some examples of these as well. So just background, there's really three types of stem cells. There's embryonic stem cells. These are pluripotent. Pluripotent means that these cells can become any cell type in the body. They can give rise to derivatives of all three cell lineages. Associated with embryonic stem cells are some ethical issues. So they have been limited in terms of the number of lines that can be used. So there have some barriers associated with that. Then there's adult stem cells. So in your body, you actually already have lots of stem cells. For example, in your skin, your skin turns over multiple times. And so you have stem cells that reside in your skin, in the basal layer of your epidermis, to be specific, that can generate new skin cells for you. So these adult cells are not pluripotent. They're already partially differentiated, so they can make limited cell types. So that's their limitation. But on the other hand, they can have a lot of good properties. And there's been a lot of work on mesenchymal stem cells, for example, that can be found in your adipocytes in your fat or in bone marrow that can be useful and have applications. And then the last, which is my favorite, are induced pluripotent stem cells. So this is from work that was done and won the Nobel Prize for, is that you can take adult cells that are already differentiated and basically de-differentiate them to become these induced pluripotent stem cells. So these are also pluripotent, just like the embryonic stem cells, and people have done comparisons. There's a lot of similarities between embryonic stem cells and induced pluripotent stem cells. They're an unlimited supply. They can just continue to replicate in their undifferentiated state. As I said, they're pluripotent, so they can differentiate into any tissue. And you can grow them from patients with diseases, with genetic risk factors, or from, quote, healthy controls. And they can be easily genetically modified as well. And there's a number of different sources to make these iPSCs. Probably the easiest source is from blood. So how do you make induced pluripotent stem cells? So this is really starting with some somatic tissue. So as I said, that can be from blood cells. You isolate the peripheral blood mononuclear cells from blood, and those are an easy source. People also take skin grafts, small skin grafts, and basically grow up fibroblasts to establish a cell line. People have isolated cells from urine. People have isolated cells from hair, and from the keratinocytes. And these are all potential sources to make the induced pluripotent stem cells. So you reprogram, you take those cells that you've isolated, you reprogram them with some transcription factors. You can expand and characterize them. If you want to do any genetic manipulation, modification, you can do that. Otherwise, you basically passage them a number of times, and then they can be differentiated into your desired cell type and used for assays. So what are the best practices? So I tried to include this so that you get a sense of like either sources to go or things to remember when you are creating induced pluripotent stem cells. One is you want to use a non-integrating plasmid for transduction. My personal experience is that blood PBMCs are much easier to get than skin fibroblasts, but you can create them, as I said, commonly they're created from both. I have a lot of experience with using Sendai virus as a way to make iPS cells. They can be miniaturized, automated, and with practice, I mean, we essentially could convert 100% of PBMCs from different ages, from different people with diseases and people who are healthy. It's a very reliable method for making iPSCs. It takes some time, so you have to count on 2 to 3 months with multiple passages. So in the case of Sendai virus, you want to do enough passages so that you get rid of the virus that was used for the transduction and to get the stable lines. But you do want to check for any chromosomal aberrations. It's important to do that periodically, although this publication I have on the top basically shows 5 human embryonic stem cell lines that they passaged for 140 times and still saw genomic stability. So it's good to check, but you can often get passages, many passages out of a line. And then this lower paper is actually a great resource because it talks about how to handle lines that you make to maximize the chances that there won't be any chromosomal aberrations and that there won't be any issues, and that you'll be able to continue to use them for differentiating and any other applications you have. So that lower paper is a nice one. And as I said, I have other resources at the end. So understanding disease phenotype and target identification is the next section. So this is still a critical step in developing a pipeline for companies. So of interest in terms of always that search for the best and understanding specific targets that will be good as a drug target. So stem cells, as I said, can be used to model human diseases at the cellular level, and they can be also a good alternative to animal models. The problem with animal models is there's a lot of differences between them and humans, and so at least taking cells and making them from patients with certain diseases can be really good models and potentially much more predictive than some of the animal models, depending upon the therapeutic area. Of course, it is good to remember that it's still a model, and your anything that's not cell autonomous is something that you're not going to capture. But it certainly is, I think, an improvement over other cell models in which you are overexpressing a target or overexpressing something to understand its mode of action. If you can start with a human disease line, that already has a known mutation in it that's known to be causal of the disease, it's going to be more predictive and more physiological. So as I said, it can include patient stratification, and understanding the mechanisms of disease in terms of helping you to identify new targets, and also a greater understanding of the genetics that can lead to target identification. A lot of diseases have long lists of genes that are associated with that. And so if you can take patient lines and then basically stratify them and characterize them in terms of what types of mutations they have, it can be very useful. Here's an example with Parkinson's disease, a paper where they looked at alpha-synuclein, and they looked at both LRRK2 patients that had LRRK2 mutations, as well as patients that didn't, and basically had sporadic forms of Parkinson's. And they really looked at the dopaminergic neurons and looked at sort of endogenous levels of alpha-synuclein, which is the main component of the Lewy bodies that form these aggregates in the disease and develop in the cytoplasm in patients. And so they learned from this that actually LRRK2 patients exhibited sort of these diffuse cytoplasmic accumulations of alpha-synuclein that was different from the sporadic and from the control patients. And that's shown like on the lower graph, it's quantified. So this is an example where you can learn something about potentially genetic form versus a sporadic form versus control, and look at properties of the neurons that are affected, in this case, the dopaminergic neurons in Parkinson's disease. So this is an example where stem cells really helped actually to segment patient populations. So long QT syndrome is a condition in which there's an issue with repolarizing the heart after a heartbeat. It can result in an increased risk of irregular heartbeats, which can result in fainting in a mild version and even sudden death in cases. And it's known that there's different forms of long QT syndrome that are really dependent on the channel. So the most common, for example, are mutations found in the potassium channel, but there's versions of mutations in the sodium channel as well, depending upon the genetic form. And that this paper characterized, how the different forms, basically identified the different forms and looked at them with electrophysiology to characterize them. It helped define among long QT syndrome, in which the patients will have this issue in terms of fainting or seizures based on their genetic forms. This actually then tied that into their cells and how they're subtypes of the disease. And this is really important because often people who have long QT syndrome actually don't know it until they have a episode. And if that's sudden death, then, if we can identify people ahead of time with long QT syndrome so that they can be aware or on drugs to prevent this from happening, would be a big benefit. So really around phenotype, identifying, using stem cells for phenotype and target identification, you need to have, of course, multiple disease donors to be able to identify subpopulations. And this is going to depend on the disease in terms of how many. Also, you need to correlate as much of the medical information about the individuals that you're looking at their cells as possible, such as the length of disease, length they've had the disease, the age of the onset, what were their first symptoms. And so you may be able to tie some of these features in terms of the patient's medical record with the things that you see in terms of the phenotype of the cells. And so this combination of the phenotypic and functional analysis with genetic profiling is really quite compelling. And there's a lot of work around with bioinformatics to to do modeling around this as well when there's lots of large data sets to analyze. So next, in terms of using stem cells for high throughput screening, this is an example where somebody was interested in, the authors were interested in Fragile X syndrome, and so they wanted to see if they could identify compounds that could reactivate this silenced FMR gene, which is what happens in the disease. And so they did a high throughput screen using iPSCs that were derived from neuronal progenitors, and they looked at compounds that affected this FMR gene. And so you can see on the graphs below some of how they had to, some ways they had to find counter screens that they developed. So the first graph A, they looked at compounds that were autofluorescent. So that would affect the readout of their assay. So they wanted to rule out compounds that were autofluorescent. And then the middle one is an example of a compound that increases the FMR expression. And then the one on the right is cytotoxicity. So you want to make sure that that your readout isn't being affected by cytotoxicity. So these are good things to think about when you develop these screens. And I'm sure many talks up until now at this workshop have brought up the issue of counter screens so that you can hone in more quickly on the compounds that are of most interest. So here's an example of another high throughput screen for osteogenesis. So they were interested in looking for molecules that either promoted or inhibited mesenchymal stem cells, which are partially differentiated. They're in the right lineage for osteogenic differentiation. And so they, on the right-hand side is a panel of 4 graphs. The first A is alkaline phosphatase. So these are compounds that induced osteogenic differentiation, promoted osteogenesis. And C are compounds that inhibited. So they actually were able to get both types of compounds. And then they did counter screens with viability. So especially compounds that inhibit the alkaline phosphatase readout, you want to make sure that you're just not killing the cells and therefore getting a lower readout. So this is a good example of give you an idea in terms of what type of hit rate you would get. And also it's interesting to look at the spread in terms of the viability of this compound library they used. So for high throughput screening, the things to remember most are actually really a lot of the same principles as with any high content cell-based assay. You want to use a relevant cell type, the most relevant cell type that you can. And you want to also make sure that you have a homogeneous cell population, or at least a good definition of the cell types that you're measuring. And if it isn't a homogeneous population, if there's a marker to look at the cells that you're most interested in as a filter, that could be beneficial. And then also identifying possible artifacts. So you need to develop counter screens around viability. Also, if you can identify multiple measurements for the same type of property from the same screen to help confirm the mechanism of your compound hits, that would be also a benefit. So thinking a lot about counter screens for any high throughput screen, especially high content cell-based assays, is important. The nice thing about high content assays is the ability to measure multiple things at one time. So then you can look, for example, I once did an assay where we were looking at the inclusion of some aggregates within cells. And so not only can you measure sort of number of these aggregates, you could look at the size of the aggregates, you could look at the location of the aggregates. These are all parameters that help you sort out, the hits that you get at the end. So that's for high throughput screening. So now this is another area that I think is really has had a lot of interest because this is something that's always been a struggle in the pharmaceutical industry, is having good ways of predicting both cardio and hepatotoxicity testing of compounds. And so there's been a lot of efforts on, can you use stem cells that differentiated to cardiomyocytes and stem cells differentiated to hepatocytes for this toxicity testing, and will it be more predictive of clinical toxicity? So taking known compounds that are known to induce toxicity in these two areas, and looking at how predictive it is, and if there's any ones that any disconnect between them. So cardiotoxicity is responsible for about 45% of drugs being withdrawn from the market. So this is that just goes to show you how difficult it can be to predict cardiotoxicity based on animal studies, which are what largely is done, and also gives you an in vitro method to predict cardiotoxicity. So you can do the animal studies, but if you can identify any cardiotoxic compounds before they get to animals, you can save a lot of time and money. So for cardiotoxicity, drug-induced efforts are measured on contractility. You can measure the maximal rate of pressure increase of left ventricle in animals. But an in vitro method, as I said, would really allow this testing to be done much earlier in the pipeline and to test more compounds. So this has been an effort, a program that actually has been across industry and academics. And this list might be small to read, but basically you can see everybody from NIH to various companies like Fujifilm and Johnson & Johnson and Takeda, Bristol Myers Squibb, have gotten together for this comprehensive in vitro pro-arrhythmia assay development. And so really the idea is can you develop an in vitro assay to look at drug effects in which you can analyze multiple cardiac channels and use in silico reconstruction of cardiac action potentials to predict cardiotoxicity. So the idea is using human stem cell derived cardiomyocytes for this. And here's a publication that was put out in terms of validation studies and best practice recommendations if you're interested in that area. But it was really nice effort. I think it's still ongoing, and they have a website, but across multiple companies as a common need. For hepatotoxicity, typically mitochondrial depolarization is used as an early signal for hypoxic damage and oxidative stress. But by using stem cell-derived hepatocytes and JC10 staining at two different time points is an area that's growing in use. And here's a publication on high content assays using induced pluripotent stem cell derived hepatocytes. And on the right is just showing you some drugs that are known to produce hepatotoxicity, and what it looks like in one particular assay. So in terms of cytotoxicity testing, use of these in vitro assays earlier in the drug development process certainly can flag potential compounds and allow you to prioritize compounds based on the results. And hopefully not too distant future, these in vitro assays could replace the animal models, which is a big effort to try to reduce the amount of animals that are used in the drug discovery process. So patient segmentation for trials. So clinical diseases are defined based on symptoms, but a lot of times within that patient population, there's different mechanisms that lead to those symptoms for those patients. So hence, typically you do a clinical trial and you take patients with the same diagnosis, but in some cases, a drug might be found to be toxic as well as beneficial. Other cases, a drug could be toxic but not beneficial, versus a subpopulation that the drug is not toxic but not beneficial. Or best of all, in the bottom right, is a drug that's not toxic and is beneficial. So how can you identify those patients that are that the drug is not going to be toxic for and going to be beneficial, in a more meaningful way? And this will make clinical trials more efficient. And it's really the whole movement of precision medicine as well. What I see in this area is bioinformatics are really having a larger role here. There's a lot of ways bioinformatics allows, the analysis of large data sets and look at the large data sets in different ways and include genetic profiling and to be able to predict better, what responders who might respond better. One example is with amyotrophic lateral sclerosis, clinical trials have been finding that patients, if they use patients both early on in the disease as well as defined rate of progression, so that they're all following a similar path of the rate of the progression disease, that itself may produce a more homogeneous population and be able to decipher whether a drug is efficacious or not. Also, the big area around biomarkers. So we continue to have a lot of effort, and this has been throughout my career, the use of and the seeking out of biomarkers to help correlate patient outcome and response. And I think we continue to get better tools in this area as well. Imaging has had a big impact here, and people continue to use both blood and in the nervous system, CSF, potentially as biomarkers to help correlate the patient outcome and response. All right, so stem cells as therapy. So this is another area. I don't know how many people are specifically interested in this area, but it's a growing area of interest. And one thing that helped was this 21st Century Cures Act that Congress passed in the end of 2016. Basically, it authorized over $6 billion in funding, mostly to the National Institute of Health, but to other areas as well. And it helped designate this and create this regenerative medicine advanced therapy product designation as a separate designation. It allowed sponsors benefits such as guaranteed interactions with the FDA, eligibility for priority review and accelerated approval, and then also flexibility in terms of the number of clinical sites used and the possibility to use patient registry data. This is particularly important for rare diseases, and also because the cell therapy is fairly new, to allow potentially fewer patients and other sources of sort of real world evidence to add in terms of evidence for getting approval. Japan actually has been a real leader in this area. They've started an effort a little while ago that where they only require a small trial for cell therapy to demonstrate safety, and then they will give conditional approval, and then a company or sponsor has 7 years to demonstrate effectiveness to then get full approval. So this allows access to these therapies earlier in Japan, and has been attractive for a number of companies who have decided to do clinical trials in Japan for this reason. And the question is, are there going to be other countries that will follow suit or not? So there's a lot of potential areas that can benefit from stem cell therapy, and I've listed just some of them here. Everything from wound healing to heart disease to joints in terms of joint repair, since stem cells have an inherent ability to both repair and regenerate in your body. Can it be more effective, for example, as your joints get worn or injured? A number of areas in the nervous system, and I have all these areas listed here as potential. And there's probably companies around all of these working in all of these areas. So this is a paper that actually just came out around Parkinson's disease, using an animal model for Parkinson's disease, where they found that they could create dopaminergic neurons that basically engraft, and evidence of engraftment into the striatum of this animal model. And when they used a different cell type, like glutamatergic neurons, they didn't get any engraftment, and they could actually demonstrate functional connections being made as well. So applying stem cells to Parkinson's disease has been an effort that started many years ago, but people continue to sort of improve on the methods in terms of what types of cells and how best to apply them. And this is a paper on a primate model for Parkinson's disease, and then really just a little bit of the history. Over 30 years ago, basically aborted fetal midbrain neurons were being used in some Parkinson's patients for transplants. And although efficacy was demonstrated, there was a lot of variability. Some of that is believed to be in the heterogeneity of the cells that were being used. So as we get better at producing more homogeneous cells, better characterized cells, I think this will continue to improve. And there's continues to be clinical trials in this area. So this is an example where it didn't work. Animal models predicted, looked hopeful. This is using the growth factor GDNF, so cells that will secrete GDNF. So this is a different strategy, not the cells themselves, like the dopaminergic cells, as an example of the Parkinson's disease, but basically engineering cells to secrete growth factor, in this case, that should be beneficial for repair and regeneration. But unfortunately, this clinical trial failed, didn't show efficacy, and another approach using AAV also was unsuccessful. So this is we still have a ways to go in this area, is the point that I want to make. So just starting to wrap up here, just general tips for working with stem cells. You need to differentiate them to the correct cell type. And so this has been shown in a number of areas, like in the example of the early clinical trials with dopaminergic neurons. There's different types of dopaminergic neurons, depending upon where in the body you want the right type. You need a homogeneous population and a reproducible method of making them. Same thing with neurons. People found that it's important to have the right type of motor neuron. And then I think the area that is the biggest issue with stem cells right now is the state of differentiation. I think we've gotten really good at making different cell types and showing that they contain relevant markers for that cell type. But they're largely still all very embryonic stage, or very early in development stage. So they've differentiated, but they're really pretty far from a mature cell type. And this has been an issue with applying cells for heart disease. Whereas if you apply less mature heart cells, that are electrophysiologically different from the mature cells in the heart already, you can get these arrhythmias. Of course, you don't want to be producing arrhythmias to people who already have some heart disease. So considering the state of differentiation is really important. In terms of characterization, just doing immunohistochemistry and showing the marker is not enough. It's better, you need a gene transcription signature. You need to look at functional endpoints, for example, transmitter release, electrical activity. A lot of times, specific targets have a early form of that target that then with maturity and with adulthood in that tissue goes through some modifications. So understanding that is important. You need to look at lots of patients. So there's a lot of heterogeneity. We're all different from each other, and being able to basically do the numbers so that you can understand that heterogeneity better is going to be a big strength. You should expect to observe some variation, both in severity and phenotype. I mean, this is the same kind of variation that you see in patients. So, so being able to reproduce that in a dish has a lot of value to it. And then the possibility of being able to do gene editing, to produce the controls, so that you can identify what might be contributing, to the phenotypes that you see, is also a good tool to have. This is just an example of the limitation. So I mentioned, with cardiomyocytes, how heart cells that you differentiate from induced pluripotent stem cells versus taking primary heart cells from adult. And this is the list of around morphology, around contractility, around metabolism. You see changes in all of these, and often you can pinpoint what you see in the iPSC-derived cardiomyocytes is really found in early stages of differentiation during development, and then you get changes in expression levels and changes in terms of morphology and the sarcomere structure that you start to see in greater maturity. And there's a lot of effort actually in a few places to look for how you can facilitate that greater differentiation and the greater maturity, so that the cells can be more useful. So this is the slide I mentioned before that has a lot of useful links to get more information. STEMbook is something that I have relied on a lot. It has a lot of protocols, and it has a lot of, in terms of best practices around protocols, and covers a lot of area in terms of information. Also, CIRM, the California Institute of Regenerative Medicine, has a website, as I list, Ensemble, Eurostem Cell. Harvard Stem Cell Institute also has some good references, LifeMap, ISSCR. International Society for Stem Cell Research is the major organization that holds meetings every year, has a journal, has a lot of good stuff on their website, et cetera. And so these are all good resources to explore. So I just want to end with, what I started with. I hope that I've been able to convince you that, stem cells can provide an important tool in our toolbox for drug discovery. They're human cells. They haven't been immortalized. They're not derived from a tumor. So they're uniquely have some human biochemistry that you don't see in other sources, other than potentially primary cells, which can often be hard to get and hard to grow. They can be used in multiple areas of drug discovery and drug development and clinical trials, and I hope I've given you some good examples of that. You can manipulate them. So even though they can be harder to grow than your typical cell line, I think you can, for those who are skilled in cell biology, you can quickly learn how to handle them appropriately. There's a lot of case studies. They can be therapeutics themselves, and they can help accelerate the process of drug discovery. And I thank you, and I'd be more than happy to take any questions you might have.
Thank you very much, Marcy. You do have one question, so maybe we can tackle it really fast because we're running late on time. The question is, are there specific differences between properties of iPSCs versus natural sources like embryonic or adult?
Yeah, so definitely there's differences between adult stem cells because they're already partially differentiated. They're not pluripotent. People have done comparisons between human embryonic stem cells and iPS cells, and there's a lot of similarities. So I think I personally have used differentiation protocols that have been used in both and find that they're, one works well for embryonic stem cells. It also works well for iPSCs. So
Okay, go ahead.
that's the short answer. I'm sure if you look hard enough, like anything else, you'll find, some differences when you look at transcription profiles and look more in depth. But, figuring out the relevance there, but I think they're a good substitute for embryonic stem cells.
Thank you very much, Marcy, for a very informative presentation.